everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and what we were discussing about the biomolecule, and in this context, so far what we have discussed about the nucleic acids and we have also discussed about the carbohydrates. In our previous lecture, we were discussing about the structure as well as the function of the carbohydrates and how the carbohydrates are being utilized in the different types of metabolic pathways so that they can be able to utilize to produce the energy. Now in today's lecture, we are going to discuss about the another biomolecule, and that biomolecule is the lipids. So as you can see here is that the lipid biomolecules are required for the energy productions, and lipids are as you can see that when we were discussing about the Krebs cycles, the lipids are being run through the beta oxidations, and that's how they are actually producing the acetyl CoA, and these acetyl CoA are actually entering into the Krebs cycle to produce the energy. So the first question comes is, what is lipid and what is the structure of a typical lipid? So what is lipid? So the first question is, what is lipid? And the lipids are the heterogeneous group of naturally occurring compounds including the fat, oils, steroid, waxes and the related compound that are related more by their physical than by their chemical properties. They have the two common properties. So lipids are relatively insoluble in water and they are soluble in the non-polar solvents such as ether and chloroform. So what you see here is that a typical lipid has the two component. One, it has a backbone, and then it has the side chains. So these side chains are backbone is mostly been present as the glycerol. So it has the glycerol backbone or the alcohol backbone, and then it also has the fatty acids. So you can have the three chains of the fatty acids. So this is the chain number 1, so this is number 2, and this is the number 3. And this acid group is actually making a linkage with the alcohol what is present onto the glycerol, and that's how it is actually forming a lipid molecule. So what you see here is the 3 fatty acids are coupled with the 3 alcohol group what is present onto the glycerol. So let's understand these backbone and the side chain first. So what is glycerol? So glycerol, which is also known as the glycerine, is a trihydroxy alcohol. So what you see here is the chemical formula of glycerol. So CH2OH, CHOH and CH2OH. Trihydroxy alcohol, as it contains the 3 hydroxyl group. It can be obtained from the diet, from the lipolysis of fat in the adipose tissue and from the glycolysis. It can be utilized for the synthesis of the triglycerol, phospholipids, glucose, or can be oxidized to produce the energy. So glycerol can directly be taken up into the different types of metabolic pathways, and that can be utilized for the production of the triglycerides, phospholipids, and even it can be converted into the glucose to produce the energy. It is used as a solvent in the preparation of the drug as well as the cosmetic industry, and the nitroglycerin is always being produced from the glycerol, and that is being used as a vasodilator. What is meant by the vasodilator is that it is actually going to dilate the blood vessels. So you can imagine that if somebody is having a blockage, right, if somebody is having the heart related or cardiovascular diseases, their blood vessels are actually going to narrow down, right. So their blood vessel is actually going to have the internal clots, and because of that they will get narrowed down. So if you provide the nitroglycerin, it is actually going to dilate these blood vessels, and that's how the blood flow is actually going to be more in these blood vessels, and that is actually going to give us some kind of relief to these patients. Then we talk about the fatty acids. So fatty acids are the aliphatic carboxylic acids. They have the general formula as RCH2N COOH. They occurs mainly as a ester in the case of natural fats and oil, but do occur in a unsteric form as the free fatty acid as transported form found into the plasma. So these fatty acids are either being present in the esterified form, or they are also present as a free fatty acids because they are being transported that way into the plasma. So when we take the food, right, the food is actually a complex lipid molecule, and that complex lipid is actually going to be digested by the lipase, and that lipase enzyme is actually going to degrade the fat into the free fatty acids, and these fatty acids are then being absorbed by the alimentary canal, from the alimentary canal, and that's how they will transport it into the plasma. Fatty acids that occur in the natural fats are usually the straight chain derivatives containing a even number of carbon atoms. They may be saturated or the unsaturated. They may be saturated, which means they will contain the no double bonds, or they can be unsaturated, which may contain the two or more double bonds. Fatty acids can be of many types. According to the nature of the hydrophobic chain, it can be saturated or the unsaturated, or the branch chain fatty acids, or the substituted fatty acids. Saturated fatty acids do not contain the double bond, where the unsaturated fatty acids contain the double bonds. Uh, Saturated fatty acids, these are the examples of the saturated fatty acids. So saturated fatty acid can be envisaged as a based on the acetic acid as the first member of the series, in which the CH2 is progressively added at the terminal CH3 and COOH. So you can imagine that the acetic acid is the first saturated fatty acid. So if the number of carbon atom is 2, then it is going to be the acetic acid or the ethanoic acid. The formula is CH3COOH. Now on this, if you add another molecule of CH2, then it is actually going to produce the butyric acid. So what you see here is the 4 carbon. So 4 carbon is butyric acid or the butanoic acid, and it is actually going to be CH3, CH2, 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 COH. So accordingly, it could be 6 carbon, 8 carbon, 10 carbon, and so. So fatty acids in the biological system usually contain the even number of carbon atom, which means it can be 2, 4, 6, 8, 10, and so on. And typically 14 to carbon atoms are present into the saturated fatty acids. The hydrocarbon chain is almost invariably unbranched in the animal fat. A few branch chain fatty acids are also being found into the plants and the animal sources. Then we had talked about the unsaturated fatty acids. As the name suggests, the unsaturated fatty acid means it is actually going to have the one or more the double bonds, right? And the unsaturated fatty acid could be a monosaturated fatty acid or to the polysaturated fatty acids. Monosaturated fatty acid is going to contain either only the one double bond, and they are going to be called as monoethanoids or the monoenoic, whereas the polysaturated fatty acids are containing two or more double bonds. The configuration of the double bond in most fatty acid is cis, so there is no trans fat double bonds. The double bond in polysaturated fatty are separated by at least one methylene groups. The systematic name for a fatty acid is derived from the name of its parent hydrocarbon by the substitution of oic for the final E. For example, if you have the C18 saturated fatty acid, it is called as the octadecanoic acid because the parent hydrocarbon is octadecane. So in the fatty acid, the last E can be just replaced with the oic. So for example, the C18 saturated fatty acid is known as the octadecanoic acid because the parent hydrocarbon is octadecane. Similarly, the C18 with double bond, it is called as the octadecanoic acid. With the two double bonds, it is called as the octadecanoic acid, and with the three double bonds, it is going to be called as octadecatrienoic acid. Now let's talk about the classification of the lipids. So lipids, as I said, it's going to have the the backbone, which is going to be alcohol backbone, and then it is also going to have the different types of fatty acids. So these lipids could be of three different types. It could be the simple lipids, it could be the compound lipids, or it could be the derived lipids. In the simple lipids, you can have the examples are like fat, oil, and waxes, where you are going to have the glycerol as a backbone, and then you are going to have the fatty acids. In the compound lipids, you can have the phospholipids, glycolipids, and the sulfolipids. So here you are going to have the some kind of derivatizations onto the side chain as well as onto the backbone, and that's why it is called as the compound lipids. And then you have the derived lipid. These are the special class of lipids where you are going to have the modification in terms of the backbone as well as the side chain. So it's going to be alcohol, it's going to be a fatty acid, or it can be going to be steroid or isopenoids. So depending on the backbone, the derived fatty lipids could be of different types. So let's first understand about the simple lipids. So simple lipids are the ester of the fatty acid with the various alcohol. You have the two examples, one is fat, the other one is called as the waxes. The fat, the ester of the fatty acid with glycerol. The oils are fat in the liquid state. So oil is the fat which is actually being present in the liquid state, whereas the fat could be the solid. Then we have the waxes, the ester of the fatty acid with the high molecular weight monohydric alcohol, that is called as the waxes. One of the class example of the fat is the triacylglyceride. So triglyceride is one of the fat molecule, right? And the triglycerides are the ester of the glycerol and the fatty acids. It could be the monotriglycerides or the ditriglycerides. So mono or diacylglycerol, whereas one or two fatty acids are steric with the glycerol and found in the tissue. They are naturally occurring fat, and oils are mix of the triglycerides. They are insoluble in water. Their specific gravity is less than 1, and they are consequently all fats float into the water, which means they are very light. So they are light, and because of that they are actually going to be float onto the water. Oils are liquids at 20 degrees. They contain the high proportion of the unsaturated fatty acids. Fats are solid at the room temperature and contain the saturated long chain fatty acids. So if it is containing the unsaturated fatty acid, it is going to be it is going to be liquid, and that is going to be called as oil. If it is containing the saturated fatty acid, then it is actually going to be solid. So and that solid is going to be called as the fat. The triglycerides are the storage form of the energy in the adipose tissues. What is the function of the triglycerides? It is a major lipid in the body and the diet. It stores fat, provide about the 60% of the fat's resting energy needs. It is produced for the insulation and the protection, and it is actually also be required for the carrier of the fat soluble compounds, and it is also been sensory qualities like the flavor as well as the texture. Apart from that, we can also discuss about the wax, right? So they are the ester of the higher fatty acids with the higher monohydroxy aliphatic alcohols. They are very long straight chain of the 60 to 100 carbon atoms. They can take up the water without getting dissolved in it, and they are used as a basis for the preparation of the cosmetics, ointments, polishes, lubricants, and the candles. So you know that the wax is all used for the production of the candles. In nature, they are found on the surfaces of the plant and as well as the insects. So wax in the plant, or in the case of wax insects, they are actually been utilizing the waxes only to repel the water molecules. Now let's talk about the compound lipids. So compound lipid, the ester of the fatty acid containing group in addition to the alcohol and fatty acids. So in this case you have the phospholipids. So lipid containing in addition to fatty acid and alcohol a phosphoric acid residue. They frequently have the nitrogen containing bases and the other substituent. For example, we have the glycerophospholipids or the sphingophospholipids. So in the phospholipids, instead of in addition to the alcohol base or the fatty acid, you are also going to have the phosphoric acid as the additional molecules. Then we can have the glycolipids. So lipids containing a fatty acid, sphingosine, and the carbohydrates are called as the glycolipids. And then we have the other complex lipids such as the sulfolipids and aminolipids. Lipoprotein are also found under this category. So let's talk about first the phospholipids. So in a compound lipid, I have taken an example of the phospholipids. So based on the nature of alcohol, it could be of the glycerophospholipids or the sphingospholipids. In the glycerophospholipids, you have the glycerol as the alcohol group. So what you see here is this is the backbone, right? This is the backbone what you see, and onto the backbone, the first carbon as well as the second carbon is esterified with the fatty acids, whereas the third oxygen is being there is a phosphate group which is attached. Onto this phosphate group you can have the different types of functional group which is going to be attached, and depending on the functional group you can have the different types of phospholipids. You can have the phosphatidylcholine. If the X is being replaced by the choline, then it is going to be called as the phosphatidylcholine. If it is replaced by the ethylamine, then it is going to be called as ethylamine. So this is called as PC, this is called as PE. Similarly, you can have the phosphatidylserine, you can have the phosphatidylinositol, phosphatidic acid, cardiolipin, plasmogen, platelet activating factor, as well as the phosphatidylglycerol. Similarly, you can have the sphingospholipids. So in the sphingosphospholipid, the sphingolol is the alcohol group, for example, the sphingomyelin. So in this case, you are actually changing the backbone also. So these are the different types of lipid molecules what is present, right? So you can have the different types of functional group, the X what is can be, and that's how you can have the different types of phospholipids. Then we have the sphingospholipids. One of the classical example is sphingomyelin. So sphingomyelin is where you can actually have the sphingosine which is being attached to the one of the carbon, right? So sphingolipids, and on this you can also have the X molecule, and this X, based on this X you can have the different types of sphingomyelin. So the backbone is sphingosine instead of the glycerol in the case of sphingomyelin or sphingospholipids. A long chain fatty acid is attached to the amino group of the sphingosine to form the ceramide. The alcohol group at the carbon 1 of the sphingosine is esterified to the phosphocholine, producing the sphingomyelin. So if you have the phosphocholine, then it is going to called as a sphingomyelin. And the sphingomyelin is an important component of the myelin of the nerve fibers. Then we have to discuss about the function of the phospholipids. So phospholipids are the major component of the cell membrane, mitochondrial membrane, as well as the lipoprotein. They participate in the lipid absorption and transportation from the intestine. They play an important role in the blood coagulation like the phosphatidylserine, right? And the required for the enzyme action, especially in the mitochondrial electron transport chain. Choline act as a lipotropic agents, and then the membrane phospholipid act as a source of arachidonic acid. It acts as a reservoir of the secondary messengers such as the phosphatidylinositol. So this phosphatidylinositol is important for the calcium signaling. In the case of the many type of responses, there is a phosphatidylcholine which is actually going to be responsible for the release of calcium from the endoplasmic reticulum, and that's how it is actually going to induce the calcium signaling. It acts as a cofactor for the activity of the lipoprotein lipase, and the phospholipid of the myelin sheath provide the insulation around the nerve fibers, and the dipalmitoyl lecithin act as a surfactant, which means surfactant means it is actually going to function as the detergent. So these are the things we have discussed about the phospholipids. Then we can also discuss about the glycolipids. Glycolipids differ from the sphingomyelin in that they do not contain the phosphoric acid, and the polar head function is provided by the monosaccharides or the oligosaccharide attached directly to the ceramide by an O-glycosidic linkages. And so this is what you see. and the number and the type of carbohydrate moieties present determine the type of the sphingolipids. There are two types of the glycolipids, neutral glycosphingolipids or the acidic glycosphingolipids. What is the function of the glycolipids? They occur particularly in the outer leaflet of the plasma membrane, where they contribute to the cell surface carbohydrates. They act as a cell surface receptor for the various types of hormones and the growth factors. They play important role in cellular interaction, growth and development, and they are source of the blood group antigens and the various embryonic antigens, and the G1 act as a receptor for the cholotoxin in the human intestine. Then we'll talk about the sulfolipids. Sulfolipids are the another compound lipids. They are cerebosides that contain the sulfated galactosyl residues. They are negatively charged at the physiological pH, and they are found predominantly in the nerve tissues and the kidney. The failure of the degradation caused them to accumulate into the nerve tissues. So lipid is actually being digested or being metabolized by the different types of pathways, right? And if that doesn't happen, then the lipid is actually going to be stored into the different types of organs, right? For example, the nervous tissues, livers, and all other kinds of tissues. And as a result, the lipid storage is actually going to be responsible for the different types of diseases, because lipid has to be utilized for the energy production or other kinds of functions. If it doesn't happen, then the huge quantity of lipid is actually going to be accumulate into the cell, and as a result it is actually going to cause the different types of diseases. For example, it can cause the Tay-Sachs disease. So Tay-Sachs disease is happening because there is a deficiency of hexokinase enzyme, and because of this there will be an accumulation of the GM2 ganglioside, and it is actually going to be responsible for the mental retardations, blindness and the muscular weakness. Similarly, you can have the Fabry diseases, you can have the metachromic leukotriopathy, and there are other kinds of fats what is going to be stored into the different types of parts of the body. Then we have the some more like Gaucher's Gauch disease, the Gaucher disease, where you have the beta galactosidase, the enzyme which is going to be absent, and because of that the lipid glucosyl ceramide is going to be accumulated, and it is responsible for the enlarged liver and the spleen and the origin of the long bones and the mental retardation in the infants. Similarly, you have the Niemann-Pick disease or the Farber diseases, and that is there you can actually have the different types of fats what is going to be stored, and mostly it is responsible for the different types of mental as well as the other abnormalities into the different types of organs. So this is all about the simple lipids as well as the compound lipids, and then talk about the derived lipids. So within the derived lipid you have the special type of lipids. So within the derived lipid I have taken an example of only one one lipids, which is called as the cholesterol. So cholesterol is a different structure and the different biochemical properties, and does not follow the rule where it has a backbone and then fatty acid and all that. So, but the cholesterol is occurs both in the free form and as well as in the ester forms. In cholesterol ester, the hydroxyl group on the position 3 is esterified with a long chain fatty acids. The cholesterol esters are formed by the transfer of acyl group by the acyl transferases. In plasma, both the forms are transported in the form of the lipoproteins. The plasma low density lipoprotein or the LDL is the vehicle of the uptake of cholesterol and the cholesterol ester into the many tissues. The free cholesterol is removed from the tissue by the HDL molecules, and it is transported into a layer where it is eliminated from the body either unchanged or after conversion into the bile acid in the process known as the reverse cholesterol transports. So this is the structure of the cholesterol. What you see here, it has the different types of rings, and then it has the this chain, right? So what is the function of the cholesterol? So cholesterol is widely distributed in all cells of the body, but particularly in the nervous tissue. It is a major constituent of the plasma membrane and of the plasma lipoproteins. It is synthesized in many tissue from the acetyl-CoA, and it is the precursor of all other steroids in the body, including the corticosteroids, sex hormones, bile acid, and the vitamin D. So, major function of the cholesterol is that it is a major constituent of the lipid what is present into the plasma membrane, and it is also being responsible for the biosynthetic pathway or the biosynthesis of the different types of the corticosteroids or as well as the sex hormones. It is also responsible for the biosynthesis of the bile acid and the vitamin D3. Cholesterol is a major constituent of the gallstone, right? So you know that there are stones which are being formed into different organs, whether the stones are found into the gallstones or the onto the kidney, right? So the cholesterol is a major constituent of the gallstones. Its chief role in the pathogenic processes is a factor in the genesis of a disease which is called as the atherosclerosis of the vital arteries, causing the cerebrovascular, coronary and peripheral vascular diseases. So cholesterol is an important lipid, and it can be transported into the blood by the help of the LDL, and whereas the HDL is a lipoprotein, high density lipoprotein, which actually can carry the cholesterol or the high amount of cholesterol into the blood, into the liver, and within the liver then it is going to be metabolized to produce the bile salts, and these bile salts are being utilized to help in the lipid digestion and other kinds of things. So this is all about the lipids. What we have discussed so far, we have discussed about the general structure of the lipids, where we have discussed about that the lipid is made up of a alcohol backbone and the fatty acids. This alcohol backbone could be of different types, and depending on the alcohol group, the lipids could be of different types. Similarly, the side chain or the fatty acids also could be of different types. The fatty acid could be saturated fatty acids or to the unsaturated fatty acids, and depending on the different types of functional groups what is present onto the alcohol backbone, the lipids could be of the different types like the phospholipids or glycerolipids or the sulfolipids. And then we also have the special class of lipids where we have the cholesterol and the other lipids. So with this, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss some more biomolecules which are important for the cellular functions. So with this, I would like to conclude my lecture here. Thank you.